So, we will continue our discussion on the scaffold. So, scaffold, if I may recall, this is a 3-dimensional interconnected porous structure. So, one of the things that I have mentioned a couple of times during the last module is the interconnected porosity. So, this is interconnected porosity in 3D space. So, that is one of the important aspects of a biomaterial scaffold. So, there are two type of things that I have mentioned in this scaffold definition. One is that micro porosity and one is that macro porosity. So, micro porosity is like 1 to 10 micron and macro porosity is like more than 40 to 100 micron. So, there is no distinct demarcation in terms of the length scale of the porosity, but it is quite widely accepted in the biomaterials literature that micro porosity is like 1 to 10 micron and macro porosity is like more than 40 micron, 100 micron and so on. So, micro porosity is useful for initial cell attachment and cell adhesion and migration. Macro porosity is more useful for bone ingrowth as well as angiogenesis, vascularization, for all those things that you need a little bit so that bunch of blood vessels can grow inside this tissue structure and so on. So, it also exerts certain mechanical and biological influences to modify cell behavior. So, typically the term scaffold is restricted to 3-dimensional porous constructs with interconnected porosity, and here is like 10 to 100 micron and facilitate tissue ingrowth and reduce limitations due to diffusion of nutrients and so on. So, morphology and porosity are of primary importance to scaffolds, whereas mechanical properties of secondary importance. So, tissue engineering, this is also this is very established field currently, and tissue engineering is defined as a creation of new tissue for the therapeutic reconstruction of the human body by the deliberate and controlled stimulation of selected target cells through a systematic combination of molecular and mechanical signals. So, molecular signals means you can add growth factors along with the cells for the target specific in a target specific biomedical applications. Mechanical signals means that scaffold itself has a certain elastic stiffness property. So, that elastic stiffness can also help in that mechanical signal transduction mechanisms. So, implant also has been sufficiently mentioned in the last module, and it is a general term used for describe any object that may be placed in direct contact with living tissue. And for implants, that mechanical properties like strength properties, like elastic modulus properties, these are like more of importance. But one thing I have mentioned and I must reiterate here, that implants must have uncompromised biocompatibility property. That means that biocompatibility property is equally important, as important is the strength and elastic modulus. Most of the metallic materials for structural biomedical implant applications are used like Ti6Al4b, titanium 6%, aluminum 4%, vanadium, stainless steel 316L, like that austenitic stainless steel grade with 18:8 chromium nickel ratio, and also it is low carbon grade. L stands for low carbon grade. So, this low carbon grade stainless steel 316 also has reasonably good corrosion resistance. Those are like popular example of the implants, whereas these scaffolds, that more of the polymeric scaffolds are quite widely used. Polymeric as well as metallic scaffolds, they are quite polymeric and ceramic scaffolds, they are quite widely used because you can make some foamy structure or porous constructs of this polymeric, and many of the polymeric materials, many of them are biodegradable in nature, so that they can degrade inside the body and during this long term exposure to physiological medium or physiological environment, and as a result these biodegradable polymers have attracted lot of attention in the biomedical community. So, while scaffolds too may be considered as implants, but all implants are not scaffolds. So, this is one of the important concept that you must remember, and conceptually a porous scaffold and non-porous implant would have different level of interactions with the host tissues. For example, a porous scaffold will promote tissue ingrowth and tissue regeneration, while an implant is expected to augment lost tissue function. So, although they have they are expected to have very good biocompatibility property for their use in medical applications, but however, they have different level of interactions with the living systems. So, in this context, another term which is quite relevant, and that is why I am introducing this term, that is called vascularization. So, it is a medical term used to describe the presence of vessels in a tissue, and in animals this generally denotes the presence of blood vessels. So, vascularization is one of the key challenges in tissue engineering. So, any scaffolds that you want to develop or any scaffolds that you fabricate in laboratory, it must have vascularization property, and this is essentially introduced by the fact that above a critical pore size, mass and oxygen transport in scaffolds is severely limited by diffusion, and leading to the formation of necrotic core in the scaffold that actually facilitates the dead cell formation. So, though pore architecture in 3 dimension, and this pore architecture in 3 dimension, it is mostly determined, it is mostly used, it mostly revealed by a technique which is known as microcomputed tomography. So, microcomputed tomography gives you both qualitative and quantitative results of the porous architecture in a scaffold materials. Another important thing like vascularization is angiogenesis or new angiogenesis. It is a term used to describe the formation of new blood vessels, and new blood vessels is very important, and this is very common in tumors and to a lesser extent tissues around implants. So, this here again the pore architecture significantly influences the angiogenesis. So, therefore, both the vascularization property as well as angiogenesis property, these two important property are determined by the 3 dimensional pore architecture in a tissue engineering scaffolds. Okay. So, having defined both the implant as well as tissue, because these two terms are, as I said in the last module, they are used interchangeably, and quite many of the people they think that they are synonymous words, implant and scaffolds, but they are not actually. That is why I have spent so much effort to explain you that why the scaffolds is different from implant. Now, let me discuss in little bit more details on another important thing is the blood compatibility. So, which has been mentioned earlier, but let me spend some more time just to explain that blood compatibility property. Now, why blood compatibility is so important, or why I am emphasizing so much on blood compatibility, because irrespective of whether it is a scaffold or it is a implant material, each of the synthetic material when implanted in an animal, they will interact with the blood with the blood which is actually a tissue. So, blood is actually a tissue, and because it contains lot of cells, I will explain to you in soon, and therefore, the compatibility of blood with a synthetic material is equally important. So, definition wise, it involves the study of compatibility of synthetic material with blood and blood cells, and this is there's two terms is there I have mentioned. One is hematology and one is coagulation. Hematology is actually study of red and white blood cells and their quantification in blood, hemolysis and products of leukocyte activation, whereas coagulation is indicated by platelet addition. So, platelet addition is important aspect that one is to worry about while evaluating the blood compatibility and fibrinogen adsorption on a material substrate. So, an ideal hemocompatible material, which is may be very difficult to find out, an ideal hemocompatible material should not cause platelet adhesion, and should be non-thrombogenic in nature. So, this is one of the things that in terms of hemocompatibility, material should not facilitate the platelet adhesion and also should be non-thrombogenic in nature, or it should not disturb the delicate or natural balance of RBC and WBC. So, all these factors there. So, This other definition of hemocompatibility is that it determines whether materials used in the device and the device itself, when operated at its maximum conditions, does not cause excessive damage to the red blood cells. So, this is that RBC. So, it should, that RBC should not be damaged or because of the interaction with the synthetic material. So, little bit introduction of the blood as a tissue. So, this is this blood, in case of blood you have this yellow colored fluid, this is called plasma, blood plasma, and in the blood plasma you have different type of cells. These are like RBCs, red blood cells, white blood cells and platelets. So, this plasma actually you can consider it as a extracellular matrix in case of these blood cells like red blood cells or white blood cells and platelets, and these are the different morphological features of these leukocytes or white blood cells. So, you have monocyte, you have eosinophil, like many times if you see that blood test report you have eosinophil count also, basophil, neutrophil and lymphocytes. So, these are like different morphology, and then certainly this morphology is quite different from that of the other animal cells that you have seen briefly in some of the other modules when I discussed in terms of the cell material interaction, so on. So, content wise blood plasma is a liquid component and it has 92% water, and it has other proteins like 7% protein like albumin, globulins and fibrinogen and other inorganic electrolytes. So, if you see this normal test tube, if you take your human blood, then you see the top part, that is the serum, and in the bottom part where lot of cells they will concentrate and they will try to settle down in the bottom part of the test tube. They are mostly comprised of the clotting factors when the blood clots like platelets and RBCs. Normally in human blood that platelet count should be 1.5 lakhs or above. If platelet count drops down quite a bit, then in case of bleeding, this bleeding will not stop in case of a patient. Okay. Now, coming to the application of hematology in the biomaterial science. So, what I will just explain to you that that this is a biomaterial, and this biomaterial, if it is exposed to the different blood elements, like that is that you have the blood plasma. Blood element means this is you have RBC, WBC, platelets and blood plasma. So, plasma is a fluid in this blood. So, there are three kind of scenario that you can immediately expect. One is that this material can be non-thrombogenic in nature, that means there will be no thrombus formation. Second scenario is that material can be thrombogenic in nature, so it can have adherent thrombus, and third one, the material is thrombogenic and non-adherent, that means one thrombus will form, but it will be actually shredded away. So, let me just show you that these scenarios little bit more with little bit more clarity. So, this is the test tube here. In the test tube you just put your sample somewhere here. Okay. One side you make your you make the test blood sample to flow and get it out of the other side. Now, when this blood elements will interact in this interaction zone with the material. So, there are a couple of scenarios that I have mentioned in the last slide is that this is the blood reaction during exposure. So, once in one case there will be no thrombus formation, this blood will simply pass through. So, that is the free of thrombus. So, that is the most ideal scenario, that is why I put a star there. The another case, second case, there is a thrombus formation, very distinct thrombus formation. Even if when the blood flow is stopped, this thrombus will remain adherent to the material substrate. So, this is, I put a circle here, this is B. So, this is what is not at all this is what it is not at all acceptable in terms of biomedical applications. The third one is that the thrombus may form, but thrombus will be taken away along with the blood. Fourth one is that thrombus will form, but it is not stable as stable like this. However, there will be lot of distributed shredded thrombus. So, some part of the thrombus will still get stuck as after the entire blood flow is stopped, and then you can see some signs of the thrombus formation. So, while nothing is ideal in our world, so this A situation is the most ideal scenario, and as long as this thrombus form it is shredded away, that is also it is okay, and the fourth one also can be accepted to some extent, but may not be in an ideal scenario. So, so this is just an example of the thrombus formation. There is two material which are used or which are potential use in the heart valve applications. One is commercial pure titanium, CP stands for commercially pure titanium, and another one you have a diamond like carbon coating, DLC stands for diamond like carbon coating, and diamond like carbon coated commercially pure titanium. After the standard hemocompatibility testing, in commercial pure titanium you can see large, huge, significant thrombus formation. But in the DLC coating, this thrombus formation is significantly reduced. Now, if you do quantification, some kind of quantification, then you can see quantitatively also this thrombus formation should be statistically much less significant as compared to commercially pure titanium without any diamond like carbon coating. So, the conclusion is that diamond like carbon coating like ceramic coating is helpful to improve the hemocompatibility of these titanium based materials. Now, in terms of quantification, people have done this hemocompatibility test for 30 minutes, and they have quantified that what is the platelet composition, platelet consumption after this 30 minutes test, and they have used different type of materials, titanium 6%, aluminum 4%, vanadium, ultra accurate polyethylene, commercially pure titanium plus diamond like carbon coating, and titanium 6%, aluminum 4%, vanadium. On the top of it we have used titanium nitride coating. In one case you have diamond like carbon coating, in one case you have a titanium nitride coating. Now, after this platelet after this hemocompatibility test, now if you do some statistical analysis and you take Ti6L4V as your baseline material, then immediately you can notice that once you put titanium and nitride coating, it is significantly much less than what you have obtained with the bare material or uncoated material. Similarly, if you put titanium, commercially pure titanium and DLC coating, again DLC coating has reduced, but may not be statistically significant because these error bars may overlap, as you can see quite clearly here. So, titanium nitride coating is certainly quite useful. DLC coating also reduces the platelet consumption. So, as far as hemocompatibility is concerned, that is the reason people are trying quite consistently to use ceramic coatings on a metallic material to reduce the hemocompatibility to get a better hemocompatibility property. Now, as far as the white blood cell consumption is concerned, this WBC consumption, and here again with respect to titanium 6, aluminum 4, vanadium, people have done with the titanium nitride coating, the white blood cell consumption, there is not much significant difference in terms of diamond like carbon coating as well as the titanium nitride coating with respect to their uncoated substrate. So, in a way that ceramic coatings has shown to be quite useful, they do not disturb that white blood cell contents in the blood. At the same time, they significantly reduce the platelet consumption after the standard hemocompatibility test for 30 minutes as recommended by standard guidelines. Now, so once this cell level compatibility like cytocompatibility and blood level compatibility is established, next stage is certainly that in vivo biocompatibility or the preclinical testing in animal models, and there we have to understand that what the scientific information we would like to obtain from particularly for bone tissue engineering applications. Now, these are the 3-4 definitions that is quite useful before we get into the more details of this in vivo testing. First one is the osteointegration or osseointegration. Osteo, so anything osteo means bone, or anything osteo means that osseous structure in the body. So, osseointegration is defined as the direct structural and functional connection between ordered living bone and the surface of a load bearing implant, which is essentially non-living material, and this property is critical for the implant stability, that is a long term implant stability, and certainly it is a prerequisite for load bearing implant and long term clinical success. So, therefore, osseointegration is quite useful, it is quite important process as far as the preclinical assessment is concerned. More textbook type of definition has been given here. It is defined as a time dependent healing process, that means it is a kinetic process, and therefore, when you are to do this kind of testing in the animal models, you have to do also at a different time point. So, you have to sacrifice the animals at a different time point during your study, so that you can understand that what is the time dependent healing process that takes place when these materials are when these materials are placed in an animal, whereby clinically asymptotic rigid fixation of alloplastic materials is achieved and maintained in bone during functional loading. So, therefore, that osseointegration is to be tested when the material is to be placed at some loaded bone part, and then time dependent healing process, how this material is helping to achieve this time dependent healing process, that is kind of a central theme of this osseointegration. And it may be divided into 3 stages. One is that incorporation of woven bone formation, second one is adaptation of bone mass to load, like it can support the loading and the biomechanical loading, and third one is adaptation to bone structure to load, that is bone remodeling, and qualitatively assessed by clinical morbidity test from radiographs and qualitatively using the microcomputed tomography test and analysis of explanted bone. So, there are 2 things that one can see, that one can do that histology, just to see that how this bone formation has taken place, and in 3 dimensional you have to do micro-CT. So, micro-CT will give you more quantitative determination, or histology is more qualitative and semi quantitative in nature. The second definition is osteo induction. This is a process to induce osteogenesis. Osteogenesis essentially means that is quality described as bone cell functionality in the surrounding region of an implant. So, if you place an implant, suppose you have a long bone structure, you cut this part of the bone, then you put some implant here. So, this is a shaded region implant. So, now you see that how this cellular activity is there in the neighborhood of this synthetic material, and it is phenomena regularly seen in any type of bone healing process, and osteointegration indicates essentially recruitment of immature cells and their simultaneous stimulation to pre-osteoblast. So, therefore, to prove the osteoinduction process, you have to also see that whether the certain stages of differentiation process is taking place or not. Bone healing process, as in fractures, are primarily dependent on the osteoinduction process. So, there are 2 terms I have defined now. One is that osseointegration or osteointegration, and another one is osteoinduction. So, induction means, so you recruit immature cells and then you simultaneously stimulate them to pre-osteoblasts. And third one is that osteoconduction. Induction is this one, and osteoconduction. So, this is a phenomena, it is again with respect to the bone implants, and this signifies the bone growth on a biomaterial surface, and implant materials of low biocompatibility like copper, silver and bone cement essentially show little or no osteoconduction. Okay. So, after this term we will now start with this in vivo biocompatibility assessment, and in vivo biocompatibility assessment certain things I would like to stress upon, that is the selection of animal models, then selection of a particular defect in the animal, and how to design a study and qualitative or quantitative determination of biocompatibility. Okay. So, this so as I said before that in vivo and in vitro, maybe let me just recapitulate what I have taught you quite few modules back. So, in vivo is kind of one step ahead than in vitro in terms of the biocompatibility assessment, and particularly you have to then select the right animal model to approximate human environment, and this is a second step prior to clinical use, and this is certainly clinical for development critical for development of clinical devices. In vitro test cannot replace in vivo test simply because in vitro you cannot kind of simulate the inflammation process, immune response, and mostly in vitro tests are single cell type. As I said that unless you do core culture you cannot grow two cells, two different type of cells in a single in vitro experiment on a material, and then fourth one is the no tissue remodeling kind of that can be assessed in the in vitro. Therefore, in vivo tests are crucial and it provides certain interactions of different cell types, like different cell types are present. It also allows you to understand the effect of hormonal factors. Third, it also allows you to understand the effect of extracellular matrix. So, this is that important distinction that in vivo test, and during the in vivo long term experiments like 26 weeks or 52 weeks or 78 weeks, the physiological environment also dynamically changes. So, this dynamically changing physiological environment cannot be simulated in a simple in vitro testing in case of the simple in vitro testing in the lab scale, and that places a significant importance, that why in vivo test is really required to get more clinically relevant results. And after the in vitro test I said that one has to select a few samples for in vivo test, because in vivo test you need to go through animal ethical committee approval. So, therefore, you cannot use significantly large number of animals for your in vivo test in view of the animal welfare is concerned and in view of the strict ethical committee regulations are involved. So, therefore, you have to thoughtfully select one or two samples which can go to the in vivo test. Now, along with the control sample or along with the baseline sample, one has to further conduct the in vivo test, which I will focus in the next module.